Hello everyone, my name is Arvind Gopi. Welcome to EG Biology. Today in this video, we are going to discuss something new. It's the first episode of our Evolutionary series. Here we are trying to formulate basic questions on evolution. Let's get into the video. I'm going to start this video by explaining the evolutionary history of human immunodeficiency viruses, which causes AIDS in humans. HIV or AIDS pandemic was confirmed in the year of 1989. There were two distinct HIV viruses, HIV-1 and HIV-2. Among this, HIV-1 caused the pandemic. These HIV viruses are part of lentiviruses or a group of retroviruses which infect diverse mammals. The group of viruses which infect monkeys and primates are called SIV or simian immunodeficiency viruses. As the evolutionary history was examined, it was evident that HIV-2 was evolved from SIV which infected Futuyma. On the other hand, HIV-1, which caused the pandemic, was evolved from SIV, which infected chimpanzees. By examining the lineage, we came to know that beginning of the 20th century itself, HIV entered the population. How it entered? Humans killed chimpanzees or Futuyma for food, which were infected with SIV, which later evolved into HIV. So how do you know this? How do you examine the evolutionary history? Clearly, there is no fossil records for viruses. So how do you do it? We will try to answer these questions in this series. As far as evolution is concerned, one topic always emerges is drug resistance or antibiotic resistance. There is no doubt that antibiotics or antibacterials have great role in the human population expansion. As the drug use was increased, prevalence of resistant bacteria was also a big concern. As you know, like beta-lactam antibiotics or carbapenem antibiotics, lot of bacteria acquired the resistance evolving the gene of beta-lactamase, which can cleave the beta-lactam antibiotics. To learn more about antibiotics and its action, please click on the card. We have already done a video on that. Okay, coming into resistance, there are various ways in which bacteria acquire resistance, either by efflux pump or inactivating the antibiotics. So these resistance became prevalent after the use of these antibiotics. So the question here to ask is, do these drugs cause drug-resistant mutations? Or these mutations can be there without the drugs? Or in other words, an unexposed bacterial culture, a population, can it evolve this resistance? Also, this resistance, can it be spread among this bacterial population or from one population to another population or from one species to another species? So there are various questions. We are trying to answer these questions in the series of evolution. As we see two examples of evolution, I will use this word evolutionary arms race. For example, in the case of humans, there is constant warfare between bacteria and humans, viruses and humans. For in the case of HIV, we use AZT, that is azidothymidine, but the resistance against this drug was very prevalent. How? HIV evolved mutations to inactivate. Similarly, bacterial population evolved resistance towards antibiotics. Bacteria and viruses are engaging in warfare each other. What are the viruses infecting bacteria are called? Yes, they are known as bacteriophages. These are viruses infecting bacteria. They have a protein coat and a viral genome. They attach their tail into the surfaces of bacteria and inject their genome into the cell. Bacteria evolved resistance against these bacteriophages. How? One is through restriction enzymes. These are enzymes which can cleave the genome of viruses. At the same time, bacteria evolved mechanisms not to cut its own genome by methylating the portions of restriction sites. Another one that you might be familiar of or heard of will be CRISPR. CRISPR is an adaptive immune response of past infections from the bacteriophages. It learns the bacteriophage DNA and keeps this DNA as a molecular memory, thereby preventing further infection. So there is evolutionary arms race going on between different species. I'm going to illustrate another example. Look at the male birds of warbler species. One is a grasshopper warbler, which has a simple singing tone. At the same time, marsh warbler species, which have more complex and elaborate singing pattern, which is explained by the complex frequency spectrum. How do you explain this? Of course, there is proximate causes for this. One will examine the syrinx, the vocal apparatus of the bird, or by examining different hormonal levels, like how testosterone hormone level can affect singing pattern, and brain perceptions, brain centers. These are proximate reasons. But what is the ultimate reason? Ultimate reason is provided by examining the historical perception or evolutionary history. The singing or the complex singing pattern has evolved in males of these species because it gave an advantage for this population. For example, they were more successful in finding the mates. Also, drift away from the mates, competing mates. That could be another reason. One will get a bigger picture by examining not only the proximate cause but also the ultimate causes for this effect. As Theodosius Dobzhansky would say, nothing in biology makes sense except in the light of evolution. Given examples like antiviral resistance, antibacterial resistance, and different singing patterns in male birds, we are talking about inherited change in the properties of groups of organisms over the course of generations. Something Darwin put elegantly as descent with modification. So in the coming lecture, we are going to talk about this biological evolution and its interesting properties in detail. Stay tuned for that. Signing off, Aravind.